Hello everyone and welcome to today's webinar, Exploiting Digital Histology Approaches to Probe the Pathophysiology of SARS-CoV-2 Infection. I'm Susie Valdez of LabRoots and I will be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Leica Biosystems. To learn more, visit leicabiosystems.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. You may also submit any technical issues here as well if you have any trouble seeing or hearing the presentation. I'd now like to welcome our speakers, Kelly Hunter, Digital Pathology Analyst at the Molecular Histology Facility at the University of Birmingham, and Dr. Graham Taylor, a senior lecturer at the University of Birmingham. For a full biography of our speakers, please click on the Biography tab at the top of your screen. Welcome to both of you. Kelly, you may now begin your presentation.
Thank you very much. My name is Kelly Hunter and I work at the Birmingham Molecular Histology Facility here at the University of Birmingham, where a core facility where we provide immunohistochemistry and tissue multiplexing services as well as digital slide scanning. I'm going to introduce our facility and some of the technologies that we offer access to before my colleagues describe some work that they've been doing, including work that's come through our facility. Actually, my left arm has just this minute started aching as I have my first Pfizer vaccine this morning, which I think is going to be quite appropriate. So I wanted to start with this hand scroll diagram. Don't worry about the contents of it, but I like it because it's the first concept of the facility that we drew up a few years ago, sat together in the pub, which is such an alien concept now that I thought people would all enjoy sharing that. But effectively, the idea of this facility was to provide a set of complementary platforms that vary in either in throughput and resolution and complexity that can form a pipeline for discovery and validation of discoveries, depending on what the input question is. You can come into this at different points. This was the workflow we envisaged at the time, but I can't really imagine sat in a pub so casually at the moment. It's very strange. So obviously our workflows will start with staining, and primarily our staining platforms comprise of Leica Bond Rx autostainers. We started our Bond journey a number of years ago, and we have been collecting them ever since. We've got 4 now, and they are real workhorses. They provide simplex chromogenic stains, right the way through multiplex chromogenic and fluorescent stains, not just antibody stains, but ISH multiplexes as well, IHC-ISH combination assays, and they also provide the staining for our digital spatial profiling assay that I'll detail later. We're also currently beta testing the Lunar 4 Comet, you can see on the bottom right there, which has a staining module built in, which I'll discuss briefly later, similar to there are other similar products on the market as well. Once we've stained the slides, then generally our slides get digitized. The multiplex slides particularly need to be digitized to be interpreted. But we have a Leica bright field scanner, which we use for primarily for digitizing bright field slides for collaboration purposes, for sharing with collaborators at different sites across the country and around the world. But it's also used to digitize chromogenic stains that our researchers want to perform image analysis on. We then have the Akoya Polaris 7-color whole slide fluorescent scanner for our multiplex assays, and the Mantra 2 parallel assay development station, which is a complementary microscope. We have, as I mentioned, we're beta testing Lunar 4 Comet, which is able to perform up to 20 staining cycles automated on 4 different slides, and I'll go into that later. And the Nanostring GeoMx platform, which I'll also talk about more, which has it's a hybrid slide scanner and genomics instrument. All of that, as you can imagine, generates quite a large data challenge. So we have a number of different solutions for managing that data. We have Leica eSlide Manager, which hosts all of this, not just all of the images that come from our Peri 82 scanner, but also any other images generated from other facilities or from our collaborators who will send us the images to import into the platform to share alongside the images we're generating locally. So this gets used an awful lot. It's designed to feel like a standard pathology workflow with a slide tray on the left, and you select your slides to view them on the right. You can make some annotations to the images, as you'd expect. The data behind this is stored in the Birmingham Environment for Academic Research, which is both a research data store and a high performance computing facility, and that powers a lot of what you'll see going on in the background. This is the platform for the GeoMx, the Nanostring GeoMx instrument, which is also accessed via the web browser, and it not only stores the fluorescent scans, but also the data analysis platform, as again I'll detail shortly. But then we'll move on to data analysis, which is a key part of most of the technologies that come out of our lab. So here we're looking at a data analysis workflow from the vector scanner, a 7-color fluorescent image on the left, which has been spectrally deconvoluted, and I'll briefly touch on that workflow in a moment. And that deconvoluted image has then we've performed cell segmentation on the second image on the left. The green objects are the detected nuclei from the blue DAPI stain on the left image, and the red line around that is an estimated cell border. The next image along is a tissue segmentation image, which has separated the image into different tissue compartments. And then the final image on the right is cell phenotyping, where each of the objects from the second image, each of the cells have been assigned a phenotype represented by different colours there. We also have the Visiopharm software, which can perform the same multiplex phenotyping workflows, as you can see in the top left image there, except we also have GPU accelerated deep learning capabilities on this, so that really accelerates the tissue segmentation and the cell segmentation required at the beginning of that workflow. But it also provides other outputs, such as the heat map you can see in the top right and the t-SNE plot down in the bottom right. And here is the interface for that software. This is the analysis software from the Nanostring GeoMx. On the left, you can see the fluorescent slide scans. Down the centre, you can see the various probes that are present in the assay that can be filtered, and then on the right you can see the outputs from analyses on our combination of those two panes on the left, and I'll go into that workflow shortly. So I'm going to start by talking about the Vectra Polaris workflow. I'm not going to go into too much detail, because I know that Mike Sarace has done this exhaustingly in the past for us. But just briefly, the idea behind this technique is that we stain all of our markers on a single slide first, label them all with different fluorophores, and then we perform a single scanning step. What you can see here is a representation of all of the fluorophores on a single slide and the various overlaps. So although you can see that the peak expressions are all at discrete wavelengths, that some part of the expression for each of those colours overlap with other colours. So then the software associated with this platform, the Inform software, is used to, at any given pixel, to deconvolute how much of the intensity captured at a pixel at a given wavelength belongs to which of the markers that are co-expressing in that pixel. And so another representation of that here, if we choose Pansy K as an example, the orange bar above the circle I've just drawn shows the range of expression that has been captured at that wavelength. And then you can see that represented as signal belonging to 3 different markers. So you've got some yellow signal coming from CD68, some orange belonging to pancytokeratin, which our peak expression has come from, and then also some signal belonging to PD1. And so the software here will estimate how much of the signal belongs to each of those 3 markers and bin them together instead of in a per scan channel fashion, but they'll be binned per marker instead. So then you'll end up with a computed channel that is enriched for your marker. It's also used to remove autofluorescence from the background signal. So what you end up with here in the top left sample in image A is a spectrally deconvoluted fluorescence image. In the bottom left, we can see here a simulated bright field stain, which is sometimes easier to interpret. And then in image C, you can see the pancytokeratin has, by itself, you can view it as an individual stain because we've deconvoluted the pancytokeratin and PD1 stains that were overlapping with it, and you get a clean stain. Then you pass it through the image analysis workflow that I just described, performing tissue and cell segmentation, phenotype your cells, and then perform high-dimensional analysis and spatial analyses on the outputs from that. So the Meaningful Comet is an example of a stain image elute workflow where a lower number of markers are stained, are applied to the tissue. They're labelled, in this case, with electrophorescent dyes. You image those markers, and then you elute the whole antibody complex, and then you start again, and you keep repeating the cycles and layer the image up in staining cycles instead of staining them all at once, which means that you don't need to perform this spectral deconvolution because you can use chromogens that are sufficiently far apart on the spectrum that there's no significant overlap. And then you end up with a more high-dimensional image as this. I think this is a 27-plex image, which you can't actually view, which is where image analysis becomes essential because we can't qualitatively interpret this image. So we'll go through similar workflows we've seen before. As well as detecting protein, we do RNA species detection. These 2 images here are examples of the RNA scope assay from ACD Bio. On the left, a multiplex chromogen, and on the right, a multiplex fluorescence assay. And we also combine these assays with protein detection on the same slide. And then on the top right here, we have a diagrammatic example of the Nanostring digital spatial profiling assay, where we combine a 3-plex fluorescent stain with targeted ROI-based profiling of a larger probe panel. So the workflow here on the Nanostring is that we start off by staining our sample with a panel of either antibodies or RNA species. At the moment, you can stain panels of around about 100 antibodies or 18,000 up to 18,000 RNA species that are conjugated with barcodes attached by photocleavable links. We stain that at the same time. We stain a 4-color fluorescent stain, three probes and a nucleochem stain. We put the slide in the instrument, and the slide scans the fluorescent morphology stain, and you use this morphology image to select regions of interest that you are going to target to profile the barcoded panel that you've added. The instrument will then iterate around the ROIs you selected, irradiating with UV, cleaving those UV linkers, and releasing the barcodes into suspension, which are then aspirated off the slide and into a collection plate. We can then count those barcodes and relate the data back to which ROI they came from. So we have a hybrid 4-color fluorescence IHC image with a high-dimensional spatial genomics output. Here's an example of the ROI selection process, and as you can see, so each of these squares is an ROI, and you're able to cross-section those ROIs, microdissect them with the built-in image analysis tools on the platform, which allow you to segment on the fluorescent stain. So in this case, the blue marker is a pancytokeratin, which is highlighting the tumour in the sample, and we've used the onboard image analysis tools to segment each ROI into two, a tumour positive and a tumour negative ROI, and they will be irradiated and collected separately to have discrete tumour and tumour microenvironment measurements. If the Nanostring GeoMx I've just described is the histology lab's approach to spatial genomics, then 10x's Visium assay is the genomics approach to spatial genomics. So this involves taking a section and applying it to a slide that has a grid of RNA primers on the slide, and each spot of primers on the plate has a barcode associated with it, which tells us which spot it belonged to. We also stain it in the same way as we have before with a fluorescence morphology stain and image that. You digest the tissue onto the slide and sequence it. So then what we have is, in this case, sequencing data where each transcript can be related back to where it came from on the slide, and the output data looks something like this. So you get heat maps for expression in different areas on the slide. So just a quick summary of the platforms we have. We start off with IHC, which is really high throughput. It's good resolution. We've got single cell resolution. It's only single stains and is more of a validation tool than a discovery tool. Then we move on to Multiplex, which increases the plexity slightly to maybe 3 or 4 colours with the same resolution and fairly comparable throughput. You move on to Multiplex IF, you increase the plexity slightly at the expense of throughput, but you still maintain that resolution, and you really start increasing the discovery power. You move on to Hyperplex IHC, you lose some throughput in exchange for moving on to 40 or more targets, but that throughput really makes it a good, more relevant discovery tool. And then spatial transcriptomics, which is theoretically infinite plex. We're running a whole transcriptome at the moment on a number of different technologies, but the resolution suffers in different ways between the two technologies I've described. So you don't get quite single cell data from either of them. And then the new kid on the block is, of course, MERFISH, which answers that problem by giving you back not just cellular but actually subcellular resolution at the exchange at the cost of complexity so far, but I'm sure that will increase over time. Here's some of the companies that we work with and the sponsors that have helped us get this facility off the ground. The Molecular Histology Facility takes in both internal academic and external academic projects, but we're also establishing a new entity named Bone Tissue Analytics, which is going to be an industry-facing service contract, GCP compliant face to this service, and this is going to be part of Birmingham Precision Medicine Centre, which will include other disciplines such as genomics and immunology in a multidisciplinary industry contract kind of trial length facility. And now I'm going to pass on to my colleague, Dr. Graham Taylor, who's going to tell you what they've been doing with these technologies and many others.
Okay, thank you, Kelly. That's a great introduction to the facility, and I'm going to talk now about our project studying SARS-CoV-2 infection in tissue that's used many of the approaches that Kelly has outlined in his talk. So we studied 2 different COVID-19 related diseases, the conventional COVID-19 infection in the lungs that can unfortunately cause people to die, and also COVID-19 placentitis. So that's COVID-19 infecting pregnant women, causing them to have problems with their pregnancies, including stillbirths. So we know a lot from analysis of blood samples from COVID-19 patients about what happens in the periphery. So patients often have lymphopenia, neutrophilia. We see increases in a range of various inflammatory cytokines. We see evidence of T-cell activation, and potentially those T-cells could drive the hyperinflammatory state in the lungs that cause so many problems. And of course, there's evidence of T-cell exhaustion. But we know much less about what happens actually in the tissues of those patients. So with that in mind, we undertook a study of tissue provided to us from post-mortem investigations performed in South Wales by Dr. Esther Ude and Dr. Gareth Leopold. Patients were included onto the study if they'd had positive SARS-CoV-2 PCR swab, either in life when they were in hospital or a swab taken at post-mortem. And depending on the specific consent that was taken from the relatives of each of those individuals, we took samples from different organs, at least heart and lung in every case. And so this has been a very challenging, both in sort of technically challenging, but also in terms of the reagents that we have available to us. Of course, this is a new virus, so there's little precedent in the literature for what we're going to find. And the reagents have hitherto been untested and poorly validated. We also have additional challenges. The tissue is taken from post-mortem, so it's not only fixed, but there's always a time interval between when the patient dies and the sample is taken. That can result in autolysis and degradation. Therefore, being really robust in the validation of the reagents we've used in this study, and hopefully that will come across in the data I'll show you shortly. So let me start with the study cohort. Focused on 8 patients, 3 at the top in blue are patients that died in the community from COVID-19 after very short preceding symptomology. We also have 5 patients shown in red. These patients were admitted to hospital for quite long periods of time and unfortunately died from their disease. And so in this talk, we'll be focusing on these early versus late cases. In terms of their fundamental pathological analysis, all of those patients had evidence of diffuse alveolar damage in their lungs. As we go from the early patients to the late patients, we see increasing evidence of the proliferative phase, alveolar damage, fibrotic phase in 5 out of 8 patients. We saw evidence of organized pneumonia in 1 out of 8 patients. And looking more closely, we see thrombi in 4 out of 8 patients, evidence of secondary infection in 5 out of 8, here shown neutrophils in the lungs, and vasculitis in 3 out of the 8 patients. So Kelly's already made a great introduction to various platforms we have available to us. So let me tell you a bit about the workflow. So here's our 8 COVID-19 patients in blue on the left-hand side here. We have various controls. We have a rhinovirus pneumonia case, 3 cases of bacterial pneumonia, 10 cases of non-infectious disease over retained lung tissues, and interestingly, a case of MERS coronavirus from the early 2000s. This is another coronavirus that's somewhat similar to SARS-CoV-2. As mentioned earlier, we collected lung and heart tissues from all 8 patients and a range of other tissues such as upper airway, brain, liver, kidney, and lymph nodes from a smaller subset of the patients, depending on the consent we were given. From those samples, we've prepared tissue sections, tissue microarrays, and we've taken scrolls of tissue for processing into RNA for genomic analysis. So here's an overview of the assays we've used. We've used spatial methods here on the left, either single-plex immunohistochemistry or higher-plex Codex and Comet multiplex immunohistochemistry. On the right, we've got the molecular methods, which are quant-seq, which is the RNA transcriptome analysis. We've also successfully performed T-cell receptor sequencing on a range of samples. In the middle are some interesting hybrid methods. These include RNA scope, which detects RNA in different parts of the tissue, and the nanostring that Kelly's already outlined. And these are interesting because they combine the power of transcriptome analysis with the power of looking at where those RNA molecules are spatially localized in the tissue. To just add that for all of these spatial methods and hybrid methods, we've stained tissue successfully, either as that end result for the method or as preparation for that method using the Leica Bond system, Leica Bond Rx. So here are the outcomes from these different assays. The spatial methods give us high-dimensional phenotyping and localization of specific markers we've pre-selected within the tissue. Of course, we can detect virus in those different regions of tissue. The molecular methods are great discovery tools. They allow us to analyze what genes are expressed differentially in the different types of tissue, and also look at pathways using gene set enrichment analysis. The key point here is that we're cross-validating across all of these different methods. So the spatial methods, the hybrid methods, and the molecular methods all support one another. I want to give an overview of the immunohistochemistry. So we've used a range of techniques depending on the sample size. On the left here, we've got chromogenic immunohistochemistry using the Leica Bond Rx system. We've applied that to large areas of tissue. And on the right here, we've used the highly multiplex Akoya codec system, but on smaller tissue microarrays, this is sort of balancing the cost and the time taken to acquire different sized images. Each of these techniques have their pros and cons, and we've combined them all together in this study. We used a range of image analysis techniques to study the data. We've used conventional qualitative assessment by pathologists looking at the tissue under the microscope, and here you can see a high-plex image produced on the Akoya system, the Vectra system. And on the right here, we're looking at machine learning-based approaches which look at the data and identify patterns using complex learning algorithms such as TISNE. So here we're showing you the Genomics workflow. I won't go into too much detail, Kelly's already described it. But essentially, we stain the tissue for pan-CK, CD45, and for DNA. And then we illuminate different regions of that tissue that we select based on that staining. That cleaves off the UV cleavable linkers that are analyzed on the sequencing in Illumina platform. Let me show you some results of this work. First of all, the virus distribution in the different tissues. So after a lot of work, we've identified a number of reagents that work well, both on terms of control tissues, but also have good performance on the FFPE tissues. So these are all being stained using the Bond Rx system from Leica. And we've used here Vero cells either uninfected or infected with SARS CoV-2 virus. So here I'm showing you the results from a spike antibody, so either uninfected or infected in brown on the right-hand side. You can see very good staining. Same for the nucleocapsid antibody as well. At the bottom, we're using RNA scope. So this is this in situ hybridization method that detects particular viral transcripts, either the spike or the ORF1ab gene. You can see the red and the green staining at the bottom right of the image here, showing you positive staining of those virus-infected cells. We've used a range of approaches to identify whether a virus is positive in different tissue sections. These include immunohistochemistry, in situ hybridization using RNA-scope, and gene expression profiling by quant-seq. We've been very robust in how we score tissue, whether it's positive or negative for the presence of virus. So, for example, if we see only one immunohistochemistry marker present at the bottom of this diagram here, we would score that as negative. If we see two immunohistochemistry markers positive, but no evidence of virus by RNA-scope, we look very closely at the positive control RNA-scope probe result. If that is negative, that suggests that the RNA may be poor quality, and therefore we score that as possible. If that positive control RNA-scope is positive, it shows that the RNA was good quality, and therefore we think that the virus is unlikely to be present because it's present only or detected only at the protein level and not in the RNA level. We see the tissue positive by 2 immunohistochemistry markers and by RNA scope in situ hybridization. We score that as likely. And we see it's positive for immunohistochemistry markers, RNA scope, and we see evidence of virus by either quant-seq or genomics, gene expression profiling, and we score that as confirmed. The reason we've been very robust in the way we score virus positivity is with data like this. So these are the antibodies I showed you earlier working really well on the cell line. On the left here, you can see a bit of upper airway tissue, and you can see good staining for spike, but not nucleocapsid protein. So the spike is shown in that turquoise color. Now, some groups will score that as positive. We score that as negative. Why? Because if you take the same tissue, which has got good quality RNA, it works well on RNA scope, you can't see any transcripts from the virus in that same part of tissue. So, whether that spike protein works well on the cell lines, it works well in the lung, there are certain types of tissue where it throws up background. So one has to be very cautious in interpreting the results of virus detection in FFPE tissue, particularly from post-mortem cases. Here's another example. In the middle on the left-hand side panel, you can see a green cell that's staining positive for viral nucleocapsid. On the right, that same cell is also staining positive for spike, but we've also got other cells that are staining for spike as well. This tells us that spike and nucleocapsid should be coincident in their expression. So again, that spike alone staining doesn't look totally 100%. We've gone on to score the different tissues for the presence of counted virus-infected cells, and by that I mean virus-infected cells that express both antigens, where we get good evidence of virus positivity by RNA scope and by sequencing. In the right-hand side, you can see we can only pick up virus-positive cells in early-stage SARS-CoV-2 infection. We've never picked it up in the late stage. In controls, we never picked it up in the MERS or the other control tissues. Another interesting result is shown here. So here we're doing RNA scope and we're looking for pancytokeratin by immunohistochemistry. You can see a cell here that stains positive for spike, messenger RNA, but it's negative for pan-CK. There's another cell at the bottom there with the yellow arrow that again stains positive for spike, messenger RNA, and positive for pan-CK. So we're getting virus detection in different types of cells, and we're still looking into exactly what type of cell can be infected by SARS-CoV-2 in the lung. Our results are backed up by the Genomics gene expression profiling data. So each of these columns shows a different type of tissue. You only ever pick up SARS-CoV-2 transcripts in the Genomics data in the SARS-CoV-2 tissue, as you'd expect. And moreover, we only ever pick it up in the early infectious cases, and not the late cases. So that light brown coloured box and whisker plot shows the results for patient SARS-CoV-2A, an early case. The other two cases to the right of that are late cases, which are negative for transcripts. Now, the genomics is it gives you spatial profiling of RNA. You can see that we pick up transcripts in the alveolus at much higher levels than what we see in the blood vessel. Again, this highlights the fact that we only see virus in early, not late. So here we're looking at the quant-seq gene expression data. This is bulk sequencing data. You can see we pick up multiple virus transcripts, particularly nucleocapsid spike and ORF1AB, in early stage cases, but not in late stage. We're now going to move on to talk about the inflammatory response that's present in the lung. So here we're showing you some data acquired on the Vectra Polaris platform. The slides were stained on the Bond Rx system from Leica. You can see very nice aggregates of CD68 positive macrophages in white in the image on the left-hand side. Performed cybersort analysis on the bulk RNA sequencing that we performed using quant-seq. You can see that there's a range of different types of immune cells present in the SARS-CoV-2 tissues that are on the right-hand side of this diagram here. Using computer-aided guided analysis of the immunohistochemistry results, we've identified different regions of the tissue that we've identified as either lymphocyte enriched or macrophage enriched or neutrophil enriched. And the frequency of these different enriched areas varies depending on the type of disease, whether that's SARS-CoV-2, rhinovirus, or control tissue. We also see interesting differences in the immune cell neighborhoods between early and late. So in the bottom right here, you're looking in red, you're looking at the frequency of CD8 T cell abundance in neighborhood 2. It's much lower in the early stage cases in red compared to the late stage cases in blue. We see differences between disease and we see differences over time. Looking at the transcription data, we see evidence of collagen enhancement in SARS-CoV-2. Interesting also in MERS, but not in rhinovirus or bacterial pneumonia. We confirmed that increase in collagen by immunohistochemistry. So here we're looking at collagen 6 staining. On the left, in normal control, you can see a thin ribbon of collagen 6 around the periphery of those cells. On the right, we're looking at a SARS-CoV-2 case. You can see the thickened collagen positive staining in that lung tissue. So in summary, we can say that we see a biphasic pattern of infection and inflammation in SARS-CoV-2 infected patients. Early phase is characterized by high virus loads and an increase in infiltrating innate immune cells. Late phase by low virus loads and an increase in infiltrating adaptive immune cells with good evidence of collagen deposition. We're going to move on now to show you some very emerging recent data from our group for COVID-19 placentitis. So, a small proportion of mothers that are infected with SARS-CoV-2 unfortunately experience complications in pregnancy that can include intrauterine growth retardation, hemorrhage, premature birth, and unfortunately intrauterine death of the infant. There's various abnormalities seen in the placenta when this occurs. So here's looking at a piece of tissue from a COVID-19 positive placenta. And on the right here, you can see CD868 positive cells at the fetal-maternal interface in the placenta. Now looking at another case, it's staining here by single-plex immunohistochemistry for spike on the left and nucleocapsid on the right. You see good evidence of staining. Here we're using the Lunaphore Comet platform to perform simultaneous spike and nucleocapsid staining on the single piece of tissue, the nucleocapsid in red, spike in yellow, and you see that staining is coincident, so we're seeing double expression of spike and nucleocapsid protein in the villus trophoblast. Also showing this virus positivity by in situ hybridization using RNA scope. So you can see this first spike on the top right and ORF1AB on the bottom right of the slide here. Finally, we performed quant-seq on the placenta samples, and you can see the evidence of virus transcriptomes in a range of different cases of placentitis. In conclusion, we've shown that you can apply the range of modern immunohistochemistry and in situ hybridization and sequencing platforms on FFPE tissues. This is particularly impressive given the time between taking those samples, between death and taking those samples, and in some cases several days. We've also shown how important it is to use multiple platforms to cross-validate each other for a key result. There's a fantastic collaboration between myself, my colleague Dr. Matt Pugh, who's studying for a Ph.D. in my group, Kelly as well, of course, as part of the IHC facility, with colleagues at the University of Limerick in Ireland, Professor Paul Murray and Dr. Aina Fennell. I give special thanks to Dr. Esther Yaud and Dr. Gareth Leopold for performing the post-mortem examinations, and of course thank the families of the deceased for making available the tissue for research purposes. This work is being supported by an SFI COVID-19 Rapid Response Grant, and thank you for your attention. and we'll move to take any questions that you have. Thank you very much.
And thank you, Kelly and Graham, for that informative presentation. We will now start the live Q&A portion of the webinar. If you have any questions you'd like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen, and we'll answer as many questions as you have time for. So let's take a look. We already have some great questions coming in from our audience. Our first question: can you comment on the identity of the cells infected by virus in the lungs?
Yes, thanks. That's a great question. Predominantly epithelial cells, but we do have now evidence of macrophage infection in certain regions of the lung. So we've not seen evidence of other immune cell types being infected.
Thank you so much. And did you do RNA scope on the same tissue that you analyzed by IHC?
Yeah, that's a great question as well. Initially, we did that on serial sections. So we did the IHC on one section of tissue and the RNA scope on another. But we've recently been able to adapt the technique so we can do RNA scope and IHC on the same tissue. And that really helps with validating the staining data at the protein level, but also at the RNA level using RNA scope.
Thank you. And we have one more question from our audience. Does your facility accept applications from external institutions?
Yeah, just to add on to the last question that Graham answered, we have had some good success combining IHC and RNA scope on the same slide using fluorescence multiplex. But we've also recently been experimenting with combining the hyperplex staining with RNA scope as well, which is not fully validated yet, but it's certainly not been a bust to start with. So it's quite a delicate technique, but it does have some scope to play well with others, and that's looking really promising. Yeah, we do accept applications from other institutions. We've in the past done everything from obviously internal. Most of our projects are internal Birmingham projects, but we've worked with some external UK institutions as well, both in collaboration with UK with Birmingham academics, or just as a straight academic service. And we've also run industry-sponsored projects in collaboration with local academics, as well as straight service from industry. So we're reasonably flexible in that respect. We do have limited capacity, of course, so those sorts of projects would be assessed on complexity and merit at the time, but we are open to taking work from outside Birmingham.
Kelly, Graham, thank you so much for your presentation today. Would you like to provide any closing remarks to our audience before we go?
Yeah, no, sure. I mean, this has obviously been a huge amount of work, and it's a fantastic collaboration between ourselves and colleagues locally, nationally, and internationally. I think it really highlights the need to have access to well-validated reagents that one can use with confidence. I think that's really key. Everything builds on the reliability of your research tools, and so I think it's really important to take the time to do those validation experiments to, be confident with the results that you get at the end.
Thank you. Would you also like to comment?
Yes. I think what's been interesting from a facility perspective is seeing how quickly we were able to not just deliver, deliver assays inside an academic lab, but at a core facility level, we were able to start delivering completely new assays with new targets, both antibody and RNA targets, through a whole range of different technologies, all springing up very quickly. And I think that's really promising for the future of the field, that emerging trends there won't be a big delay between commercially available products and that we've seen in the past, and I hope to see that continue.
Thank you again, Kelly and Graham, for your time and for your important research. I'd also like to thank LabRoots and our sponsor, Leica Biosystems, for underwriting today's educational webcast. And before we go, I want to thank our audience for joining us today and for their interesting questions we did not have time for today, and those submitted during the on-demand period will be addressed by our speaker via the contact information you provided at the time of the registration. And this webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, take care. Everyone, bye-bye.